Hello, my name is Taylor Hart, and I will be presenting my project on organic carbon cycling in Santa Clara River particulate matter. I'd like to thank my mentors, Dr. Niels Hobson and Professor Ellen Druffel, for their help. Some background information on my project. There's been very little previous research on organic carbon cycling in small rivers. Almost all of it has been done on the world's large rivers, such as the Amazon or the Nile. But the difference between those large rivers and the small ones, such as the ones that we have in California, is the length, the depth, and the rate at which the water travels. This could completely change how organic carbon is cycled in these rivers, which is why we need to look at small rivers as well. I'll be talking about the Santa Clara River because it's the largest unaltered river in California, but by the world standard is still considered a small river. It is unaltered, means we will have a better insight on natural processes instead of looking at rivers that have been altered by human development. So this project also has climate change impacts. Understanding carbon in river systems increases understanding of atmospheric carbon sources and sinks. I'll be looking at two things. The first is sources. If we have a very young carbon in our samples, that means that there's modern sources, such as terrestrial plants or marine plants like algae. But if we have very old carbon, that means that our sources come from chemical or physical breakdown of rocks or from petroleum or oil. I will also be talking about cycling rate. How fast is the carbon being cycled within the small rivers? Is it going really quickly, drawn down from the atmosphere by photosynthesis and immediately respired back, or is it being buried in the sediment and kept for longer periods of time? My research question is what is the relative age, makeup, and cycling rate of particulate organic carbon, or POC samples from the Santa Clara River? My methods include carbon isotope ratios and organic compound fractionation. Carbon isotope ratios will identify the age and cycling rate of the zapped carbon. Basically, I look at ratios of different carbon isotopes, one of them being radioactive carbon or carbon-14. It decays over time. So if there's a lot of it, it means that not much time has passed, meaning it's a modern source such as the plants. But if there's very little, that means it's decayed over time, showing that it's an older source, the breakdown of rocks, petroleum or oil seeps. This will also reveal how fast it's cycled. I will then look at organic compound fractionation, where I'll look at the chemical makeup. What is the source? I'll do this by looking at the amount of lipids, acid-soluble fraction, which is amino acids and carbohydrates, and the acid-insoluble fraction. My project includes lab work, working with the Keck Carbon Cycle AMS lab at UCI to get the carbon isotope measurements and the data analysis. This project will increase our understanding of the role of small rivers in the carbon cycle. I'd like to thank my team at the Druffel Lab and also UROP for the grant money in this amazing opportunity. Thank you so much for your consideration.